Okay, now in part 2 we will be seeing how the novel technologies of sequencing replaced the Sanger sequencing and what are these technologies. So Sanger sequencing is a method, as we saw it, developed in 1977, and the sequencing is based in fact on the incorporation of a modified base that is called D-deoxynucleotide or DD-NTP. You have here an example illustrated of the difference between a normal base and the modified base. So the normal base has a 3' OH that is required for the formation of a phosphodiester bond between two nucleotides. So basically two bases of DNA would be linked thanks to the 3' OH, whereas the DD-NTP lacks this 3' OH, and this terminates the elongation of the chain. So in fact, the reaction volume contains the DNA sample that is divided into 4 separate sequencing reactions containing all 4 of the standard deoxynucleotides that are D-ATP, D-GTP, D-CTP, and D-TTP, and also the DNA polymerase. So to each reaction is added only one of the 4 DD-NTPs that terminates the chain in approximately 100 lower concentration, and when they are incorporated in the 3' end of the growing chain, they terminate the chain selectively, and this causes the extension process of DNA to be stopped. And because the process is random, then all possible sizes of DNA fragments are generated. Then they are heat denaturated and separated by their sizes on electrophoresis, on a gel. And the sequence is read accordingly. So the Sanger sequencing has been extensively used and automated by a company called Applied Biosystem. The classical shotgun sequencing was based on the Sanger sequencing method, and that strategy is still applied today. However, this time using other sequencing technologies that are called next-generation sequencing technologies, especially for large-scale genome analysis, although the Sanger method remains in wide use for smaller-scale projects, like gene sequencing, etc. So the NGS technologies are becoming these days particularly popular for biological and medical research, and they can be used to sequence short or large genomic regions and even entire genomes, full chromosomes or entire genomes. Several technologies exist that have different purposes, whether you would like to sequence an entire genome or only the exons, for example, that are the coding parts of the genome. And this is what makes NGS today a catch-all term that is used to describe a different number of modern sequencing technologies, like the Illumina sequencing, for example, and we will be seeing examples of these technologies later on. So the NGS technologies, in fact, they produce shorter reads, but many hundreds of thousands of these reads, or even millions of reads, in a relatively short time, in the order of a day now. And this results in high coverage, and this is what makes the NGS technologies really popular and obviously superior to Sanger sequencing due to the high volume of data generated and the relatively short time it takes to sequence a whole genome, although of course with the NGS technologies the assembly process is much more computationally intensive. So I prepared here kind of a summary of the different NGS technologies and the chemistry that is behind these different technologies. What we will be talking about now is indicated in red in this slide. So the technology itself that replaced the Sanger sequencing is called SBS, or sequencing by synthesis, which is the general principle that is used in the bridge PCR on a flow cell, and then pyrosequencing, which is used with the emulsion PCR on beads. So the general principle of the SBS, or sequencing by synthesis, relies on the fact that nucleotide addition is tracked as the DNA chain is copied. So basically at the same time than the DNA elongation, and this is what renders the technology completely different from the concept of sequencing with the Sanger technology, in which we had to wait for the whole process to run before having access to the sequence. So the principle is that a DNA template is immobilized and solutions of A, C, G, and T are coupled to a dye and sequentially added and removed. And then the light would be generated only when a nucleotide complements the first unpaired base, as you can see it here, so the light is emitted. And the chemiluminescent signal detected will allow the determination of the sequence of the template that is read by a camera. So this is the technology that is used in the bridge PCR. Basically what is done in the bridge PCR is that the PCR amplification occurs on a flow cell, which is a solid support in which primers will coat the surface. These primers are fixed to the surface of the support, and they are attached at their 5' end by a linker. And as a consequence, there is amplification of the products, and this amplification will occur on the surface itself, and for any DNA template that is fixed. And this DNA template will remain locally fixed near the point of origin, as well as all the PCR amplicons that have been generated from it. So this is what constitutes a cluster, and the PCR will produce then clonal clusters, each containing hundreds of copies of a single DNA. So this is the kind of technology that is used in Illumina. Now the pyrosequencing technology relies basically on the same principle of sequencing by synthesis, which is detecting light along with the elongation of DNA. And it relies, in fact, the difference is that it relies on the fact that the incorporation of the dNTP will release a pyrophosphate. And then, so we do not detect the light directly. So what is detected is the release of that pyrophosphate that would be converted into ATP in the presence of APS with an ATP sulfurylase. And this ATP will be used by a luciferase that will convert luciferin to oxyluciferin. So this conversion generates visible light in amounts that are proportional to the ATP that has been released. So if you compare these sequencing technologies to Sanger sequencing, so the SBS and the pyrosequencing, you can easily understand why they are now widely used in NGS technologies. So this is the technology that is used in 454, for example. So basically this is the emulsion PCR technology. So we have here a library that is also, as the previous one, the adapter flanked. So we will be seeing the details of the technology and why we need to have adapters. But the PCR amplification is now done not on a support, on a flat support, like the flow cell that we saw previously, but now it's in the context of a water in oil emulsion. So the PCR primer is 5' attached to a bead. The size of these beads is of a scale of a micron. We have a low template concentration, so we make sure that we have that low template in order to have most compartments containing only one bead, and that one bead will have 0 or 1 template molecule present, and this would ensure that each bead would be related to just one amplicon. And then the PCR amplicon are synthesized on the surface of the bead. And after breaking the emulsion, the beads that are bearing amplification products are selectively enriched. And then each clonally amplified bead will bear on its surface PCR products that are corresponding to the amplification of a single molecule from the library. So basically, one clonally amplified bead will bear that PCR products for a single molecule. Okay, so, and we will see examples of these NGS technologies that are using pyrosequencing or SBS and that are based on emulsion or rich PCR or other novel technologies in the third part.